Biological life operates on a strict physical paradox. The chemical reactions required to build DNA, contract muscles, or fire neurons require massive energy to begin. Under baseline laws of physics, these reactions are simply too slow to sustain a living organism. This chart shows the energy landscape of a standard chemical reaction. We are mapping free energy, or delta G, on the y-axis against reaction progress on the x-axis. The massive hill between the high-energy reactants and the low-energy products represents the activation energy. This is the physical energy barrier molecules must overcome to react. To survive, cells bypass this barrier using enzymes. These biological catalysts physically bind to the reactant molecules and stabilize that transition state, providing an alternate, easier pathway for the reaction to occur. On our reaction coordinate diagram, the presence of an enzyme drastically lowers the activation energy hill. The barrier drops and the reaction proceeds at a fraction of the original energy cost. Notice what has not changed. The starting energy of the reactants and the final energy of the products remain perfectly fixed. The reaction releases the exact same overall free energy. Enzymes possess absolutely zero ability to alter the thermodynamic equilibrium of a reaction. Their physical domain is strictly limited to kinetic acceleration. Accelerating cellular reactions by factors of 1 trillion solves the energy problem, but creates a new vulnerability. Biological speed without regulation is lethal. If every enzyme operated at maximum capacity continuously, cells would rapidly deplete their resources and destroy themselves. To maintain homeostasis, cells must dynamically fine-tune these accelerators. They use chemical signals to competitively inhibit active sites, allosterically warp enzyme shapes, and selectively halt specific metabolic pathways the moment enough product is synthesized. We cannot directly observe these molecular binding events under a standard microscope. To understand control mechanisms, we must translate invisible physical interactions into exact diagnostic mathematical geometry. By building a mathematical model of enzyme kinetics from first principles, we can map exactly how different chemical inhibitors leave distinct geometric fingerprints on a diagnostic plot. We begin by isolating the variables. In the baseline biochemical experiment, we hold the concentration of an enzyme strictly constant in a test tube, and we track the overall velocity of the reaction as we steadily add more of its target molecule, the substrate. This chart shows the initial results, plotting the reaction velocity on the y-axis against the substrate concentration on the x-axis. Initially, the velocity surges linearly. There are abundant empty active sites on the enzymes, and they readily bind the incoming substrate. But as we continue adding substrate, the curve flattens. We reach a state of enzyme saturation, where every available active site is occupied. At this point, the reaction rate hits a hard physical ceiling, which we define as Vmax, the maximum velocity. Vmax tells us the speed limit for this specific test tube, but it depends entirely on how much enzyme we put in. We need a standardized metric to measure how strongly the enzyme actually attracts the substrate, independent of the total volume. To find this, we look at exactly 1/2 of the maximum velocity. We then trace that point down to the x-axis to find the exact substrate concentration required to achieve that half-max speed. We define this specific concentration as the Michaelis constant, which we call Km. This reveals a strict inverse relationship. A low Km value indicates a high substrate affinity. The enzyme requires very little substrate to reach half saturation because it binds to it so effectively. Vmax remains a bulk measurement. It calculates the total output of every enzyme molecule in our sample working together, rather than measuring the speed of a single active site. To isolate individual speed, we divide Vmax by the total enzyme concentration. This extracts a constant called Kcat, or the turnover number. This is the absolute maximum number of substrate molecules a single enzyme active site can convert into product per second. In a cellular environment, raw processing speed must be balanced against substrate sensitivity. An enzyme that turns over molecules rapidly is ineffective if its affinity is too low to find those molecules in a crowded cytoplasm. We combine these parameters mathematically by dividing the turnover speed, Kcat, by the affinity metric, Km. The resulting Kcat over Km ratio is the universal biochemical benchmark. It accurately quantifies an enzyme's catalytic efficiency, scoring how well it performs in the competitive environment of a living cell. The Michaelis-Menten plot is theoretically sound, but it presents a practical problem. It is nearly impossible to visually diagnose subtle changes in an asymptotic hyperbolic curve. Tracking exact shifts in Vmax or Km by eyeballing a curve is highly imprecise. The mathematical solution is to apply a double reciprocal transformation. By plotting 1 over the velocity against 1 over the substrate concentration, the data points invert and snap into a perfectly straight diagonal line. This straight line crosses both axes, locking our 2 key variables into precise geometric positions. Where the line intersects the vertical y-axis, we find exactly 1 over Vmax. Tracing the line horizontally, where it crosses the x-axis, stretching into the negative quadrant, we find exactly negative 1 over Km. Because this is a reciprocal plot, we apply inverse logic to read it. As an intercept moves further away from 0 on either axis, the actual physical value it represents is getting smaller. This linear geometry allows researchers to read an enzyme's maximum velocity and affinity simultaneously. The Lineweaver Burk plot provides a precise canvas to track exactly how pharmacological inhibitors manipulate these variables. Let's test 3 distinct inhibitor scenarios, starting with a competitive inhibitor. This molecule physically blocks the active site, directly fighting the substrate for access. We can overcome this blockage simply by flooding the system with excess substrate. If substrate molecules vastly outnumber the inhibitors, the enzyme eventually reaches its original maximum velocity. Vmax is unchanged, but we require much more substrate to get there, meaning Km increases. On the Lineweaver-Burk plot, this leaves a clear geometric signature. The y-intercept representing Vmax remains anchored in place, while the x-intercept moves closer to 0, indicating that Km has increased. Next is the noncompetitive inhibitor. Instead of fighting for the active site, this molecule binds to a remote allosteric site elsewhere on the enzyme, physically altering its overall shape. Adding extra substrate cannot fix a warped active site. The overall maximum velocity of our sample drops because a portion of the enzymes are simply offline. However, the surviving unwarped enzymes behave normally, retaining their exact original affinity. Km remains the same. On our plot, the geometry reacts accordingly. This time, the x-intercept for Km remains perfectly anchored. The line tilts upward, raising the y-intercept further from 0, which proves that Vmax has decreased. Finally, we have the uncompetitive inhibitor. This molecule is highly specific. It strictly binds to the enzyme only after the substrate has already entered the active site, forming an enzyme-substrate complex. By locking the complex together, the inhibitor permanently removes active enzymes from the pool, causing Vmax to drop. At the same time, it artificially prevents the substrate from leaving, which registers as an increase in affinity, causing Km to drop as well. On the graph, the new line shifts upward and to the left, running perfectly parallel to the original. Reversible inhibitors act as distinct geometric signatures that decode exactly where and how a molecule attacks an enzyme. We now have a strict diagnostic matrix. An anchored y-intercept proves competitive inhibition. An anchored x-intercept proves noncompetitive inhibition. Parallel lines prove uncompetitive inhibition. But these elegant reversible geometries only apply if the inhibitor can actually detach. We must contrast this with the blunt force of irreversible inhibition. Irreversible inhibitors permanently modify the active site via prolonged or covalent bonds. This destroys the enzyme's capacity to function, forcing the cell to synthesize entirely new proteins to recover the pathway. This is exactly how aspirin operates. It functions as an irreversible inhibitor, permanently disabling the cyclooxygenase 1 enzyme to block the downstream synthesis of molecules that trigger inflammatory pain pathways. Mapping these kinetic interactions allows researchers and pharmacologists to rationally design drugs that target specific biological pathways based on their mathematical signatures. The Michaelis-Menten and Lineweaver-Burk models are highly effective, but they rely on a foundational assumption that enzyme active sites act entirely independently of one another. In biological reality, many critical enzymes possess multiple subunits that communicate. In a process called cooperativity, binding one substrate induces a structural shift across the entire protein, making it physically easier for the remaining subunits to bind their own targets. This chart shows the result. Instead of the simple hyperbolic ceiling we started with, cooperative enzymes yield an S-shaped sigmoidal curve. These complex allosteric multi-subunit enzymes break the linear transformation. Tracking their shifting affinities requires advanced mathematical tools, like Hill's coefficient, to measure the exact degree of cooperative binding. But the underlying principle remains intact. Whether an enzyme follows simple linear geometry or complex sigmoidal cooperativity, its evolutionary purpose is identical. It exists to strictly regulate the uncoupling of life's speed from the rigid limits of chemical thermodynamics.